So welcome to our first sensory session of 2022. As advertised, we will be talking about permissive and protective oxygen management in Sauvignon Blanc. Before we get started, though, I did want to give you an update on what's going on with the WRE and what you can expect to see coming in the next couple of months. So I'm going to go ahead and share my screen because my notes will keep me honest here. That might not work. It's not clicking over. There we go. I actually think I need to stop that a minute and do this. I'm rusty. I don't remember how to do this correctly. There we go. Hopefully you can see the screen now. We do have, I realize it's been a little bit quiet in your email box from me. I haven't been sending out a lot of newsletters, and we're getting started just a little bit later with sensory sessions, but that's really because there's been a lot going on behind the scenes with the research exchange. And I want to make sure that you know what those things are, but like I said, also let you know what's coming up in the next couple of months so you don't miss anything if you don't want to. So the first thing to let you know is that we have received wine board funding for 5 years. So many of you know that we've been generously funded by the wine board since 2016, and that's been on sort of a year-by-year grant cycle. About, I don't know, a year and a half, maybe 2 years ago, we started talking to the wine board about transitioning that funding to a little bit longer term structure that has a lot of things to do with kind of the workings of how the WRE works and how the wine board works, but they have been really gracious to work with us on that. We started to write a contract for that at the end of June. We negotiated through harvest, and starting January the 1st, we are now kind of on 5 years of funding. So first of all, if you value what the WRE does, and if this is meaningful to you, please make sure to thank your favorite wine board member. This was really something that they did that was out of the ordinary for them, and it was really wonderful for them to kind of work with us on structuring how we would be doing this for the next several years. That actually brings about a number of different things that we think are going to help us to be able to serve you better. The first of which is that we have a new exchange coordinator. So I want to introduce you to Jenna Barazzi. She's on the call with us today. She'll be sort of running the chat. She's the one that's letting you into the waiting rooms. And she's going to be handling a lot of the coordination to get our sensory sessions together, to send you your sensory sheets, to coordinate with our host sites when we're in person. She's also working on a lot of things kind of behind the scenes in data management and getting our website up to date and a number of other things that will allow me to be able to focus a little bit more on doing so we're calling me the research enologist at this point, but really to engage with you on your enology questions, to develop newsletter content, and to work on some bigger projects that I'll tell you about in just a minute. So like I said, Jenna's on the call here, and she will be coming around, when we have in-person sessions, she'll be there, and she'll probably do some sampling with me and for me. So hopefully you'll get to meet her in person sometime soon. Another thing that happened with our 5-year contract is that we will be expanding into cider. So much like in 2016, the wine board asked the research exchange to expand from the Monticello AVA to the rest of the state. Part of this new contract, they're asking us to go from just serving the winemaking community to also serving the cider community. Many of you know that the wine board funding comes from your excise tax. So anytime your wine leaves your bonded space and goes into your tax paid space, that tax eventually goes back to the wine board to administrate for research and education and marketing, and we're part of that. Well, cider also has that excise tax, and that also goes through the wine board. So their funding is partially due to cider as well. So what that will mean for us right now is that we will probably we'll continue to have wine sensory sessions as wine sensory sessions and cider sessions as cider sessions. But if you are a person who makes both wine and cider, we want to know that so we can make sure to include you on the invitations for the cider sessions as well. So if you are on the call today and you also make cider and you want to be included in that, if you want to just drop your information into the chat and say, please include me in cider, you can do that. You can also email me and let me know that way so that we can make sure to add you. Now, most of you know I don't actually know anything about cider, or I'm just learning about cider. But thankfully for our cider makers, Jocelyn Kazalka has agreed to come on as our cider content specialist. If you know Jocelyn, you know that she has a great technical knowledge about both cider and wine. She will be part-time with us at this point, so you won't see her as much as you'll see myself or Jenna, but she will be handling kind of developing the experiments and developing any of the content that has to do with cider. Because she's also a winemaker, though, it's great because it means we have one more winemaking brain thinking about our WRE projects, which is also great. Another thing that's going on with the research exchange, because again, partially because of this 5-year funding, partially because of some external funding, is we have sort of 2 larger projects that are also in the mix. You haven't seen much about these lately yet, but they will sort of be rolling out in the next couple of months. So we do have an external grant looking at using chitosan in the vineyard. That's something we're currently working with Tim Jordan and Lucy Morton to sort of design some experimentation on that. And then we will be looking for some vineyard partners on that. And you'll start to see some data hopefully rolling out from that. We've got 2 years of funding for that. So after we do our first season, we'll let you know how that is working out. And then the second one is a project looking at selective grape breeding. This is really spearheaded by Emily Pelton and Ben Jordan. But the research exchange is really acting as kind of a conduit for the funding to flow through us toward the researchers and to give a little bit of organization to that. If you haven't already heard about this project, it's very exciting. The idea is to use next generation DNA tools as markers to help us select new grape breeds. The breeding itself is traditional breeding, but from a given cross, for example, you might end up with thousands and thousands of seeds and you don't know which ones are the ones that have the traits that you want. Some of the genetic markers that have been developed through Cornell and through some other projects overseas allow us to be able to screen those to choose, which 100 seedlings do we really want to raise up. So that project is just getting it's just getting on its feet now, but you will probably be seeing a poll come through your email box, either from the VVA or the VWA, asking some things about what kind of traits you would be most interested in. That is for that particular project. If you do have more questions about how that's working, please contact Ben or Emily. And I'm also happy to answer those questions as well. So if you have questions about that, maybe stay on afterwards and we can talk some more about it. And then we obviously are starting sensory sessions for this year. So, this is the first year in a couple of years that we have had the option of whether to have our sensory sessions be virtual sessions or in-person sessions. And after we talked to as many of you as we possibly could and sort of mulled over what was the best format for that, we realized that there really are pros and cons to both. And so we will be doing some of our sessions virtually like this one, and we are planning to do some of our sessions in person as long as our public health situation allows that. I don't have a full schedule already quite yet, but we will hopefully have that out very soon. I can tell you that we will be having a virtual sensory session on February the 24th, and the email invitation for that will hit your email box about 3:15 today, I think is when it's scheduled to go out. We also have another one that's scheduled for March the 10th. And that will also be a virtual session. I do realize some of you had some difficulty in kind of finding, the email sort of had a little bit of an issue today, and really that's because of this expansion. Because we have multiple members of our team now, we can't roll everything through my email address. So the links for the sessions today came from a new email address that we will be using that will house all of our sensory forms and that sort of thing. So we're planning on basically sending out an email from my email address, which is in your book and your address book, to kind of tell you how to add that new address to your address book so that those things don't get lost in the future. So if you did have that problem today, or even if you didn't, when you do see that email in the next day or two, please do take a look at it and just make sure that your system is set up to receive emails from us. I would love to be able to circumvent all of those myself, but because everybody has a different email server, we just have to kind of troubleshoot and make sure that things work for everybody. If you do continue to have problems, please make sure that you contact me and we will try to work through that together. But either way, you should see an invitation today in the afternoon. If you don't see that invitation, then maybe we can start to look at what's going on there. Okay. Okay, so with that, let's go ahead and jump into our content for the day today. So as advertised, hopefully you could tell from the wines that you got, and I think it was even in the title, we will be talking about Sauvignon Blanc today. Many of the things we're talking about will also apply to other aromatic white wines, but today specifically, we're talking about Sauvignon Blanc. So the first thing I'd like to do is just talk a little bit about Sauvignon Blanc varietal character. So let's see. Having technical difficulties. Just a moment here. Let's see if I can find. Jenna, are you able to launch the poll?
I can try. Let's see. Yes, I can. Would you like me to launch it?
Huh?
Would you like for me to launch it?
Go ahead and, yeah, if you could go ahead and launch the poll, that would be great. So the first question I had is, just we're talking today about Sauvignon Blanc. What are some descriptors that indicate to you when a wine is Sauvignon Blanc? So the thing that arrived in your mailbox was a Sauvignon Blanc wine. How, if I hadn't told you that, how would you know? What are some of the descriptors that would mean for you that was Sauv Blanc? I've put the wine aroma wheel up here, so you might be able to see some descriptors there that remind you of Sauvignon Blanc. If you've got a descriptor that you're like, if my descriptor is not on there, then you can go ahead and type that into the chat also so that we can see what that looks like. Let me pull up the chat so I can see the chat. And if for some reason you can't see this question, that probably has to do with which version of Zoom you're running. So you can answer the question right into the chat as well if you want to. So we're just listing descriptors that indicate that to you that these wines are Sauvignon Blanc. Just another maybe 30 seconds. Okay, Jen, if you want to close the poll. And let's see. I think this is one, if I share my results, can you guys see the results here or not? Somebody could say out loud whether you can see these results.
We can't see anything.
Okay, I thought that might be the problem. So I'll go ahead and share my screen again so we can all look at these results together. So, yes, we see things like thiol, grassy, gooseberry, grapefruit, lots of grapefruit, green, asparagus, citrus, cat pea, grass, grapefruit, cat pea. You can see grapefruit, cat pea, boxwood. These things come up quite a lot. We have zippy acid, lemon, honeysuckle, fresh mowed grass, right? These are all things that we would pretty readily associate with Sauvignon Blanc. But it turns out that there's a couple of different ways that we can think about Sauvignon Blanc. These aren't the only descriptors that we can find for Sauv Blanc. There was a paper that came out in 2011 that I think illustrated this really well. What they did is they had 19 different New Zealand winemakers look at 18 different Sauvignon Blanc wines. The winemakers came up with their own set of descriptors. They agreed on them together, and then they scored these different wines for those descriptors. They also sorted the wines into categories, and they also did some chemical analysis of the wines. And it turns out these wines, there were 6 of them from France, 6 from Austria, and 6 from New Zealand. So what they found was when they kind of took all of those descriptors and all of that chemistry and put it together, they ran it through principal component analysis, which we don't have to worry about too much here. But what we do see when we plot this together, things that are closer together are more like each other, things that are further apart are more different. So if we sort of look, for example, at the French wines, those are the ones in the box here. You can see those are sort of clustering together here. We have our New Zealand wines that are clustering together over here. And we have our Austrian wines that are clustering together here. And when we look at what descriptors they came up with,
I'm not sure if I don't know if
Yeah,
it's just me, but I don't think that we're seeing what you're showing.
I'm sorry, I don't mean to be depriving you of the wonderful thing. Let me see if I can. I think it's because I went back and forth. So screen. Just have lots of technical difficulties today. Let's see. Okay, Jenna, can you see that?
Yes, we can see it now.
Now you can see it? Okay, great. Okay, so plotted them all together. Things that are closer together are more like each other. Things that are further apart are less like each other. And what they found was that the New Zealand wines indeed had a lot of these descriptors that you guys just listed, right? So they're the ones that are sort of green and fruity and grassy and herbaceous and have boxwood and gooseberry, those sorts of things. The Austrian wines were still Sauvignon Blanc, but they just had the fruitier descriptors and not as much of that green character that comes along with those descriptors. But then the French wines, you can see those are sort of clustering together, kind of not fruity and not necessarily grassy. They're like this in this different category. And I would say it doesn't mean that there aren't descriptors there. We have to remember that it was New Zealand winemakers that came up with these descriptors to begin with. So perhaps they were focusing on the descriptors for their own wines and not necessarily naming enough descriptors for the French wines. But basically, Sauvignon Blanc can come up with various different styles, in this case, depending on what the nation of origin was. Now, this could be because of the terroir itself, or it could be because of the winemaking styles that go along with this, or probably both. If we look at in just a tiny bit more detail here, if we look, we can see that sort of the descriptors we had for the New Zealand wines tend to include some of these compounds that we traditionally think about as thiols, our 3MH and our 3MHA, but then some other ones as well that we kind of think about with more green characters like hexanol and methoxypyrazine. Whereas the French wine didn't have any of those thiols at all. The Austrian wine had some esters and also some of the thiol component to it. So just thinking about that and thinking, Sauvignon Blanc doesn't have to be just the one style. It has the potential to be multiple different styles. It's in there. It just depends on what's going to come out along the way. So with that, I'd like to bring in Mattieu. So I'm going to go ahead and see how this works. Can I spotlight Mattieu? So Mattieu should come to the top there. So Mattieu, this was the experiment that we're talking about today was one that you did. So do you want to tell us a little bit about what made you want to do this experiment and what you were kind of hoping to achieve here?
Yeah, I mean, I've been making Sauvignon Blanc from a vineyard that's belonged to Childs for, it's been like the 3rd years now. And I'm also custom crushing grapes from Childs to make his own wine with the same grapes. So my style is being a bit more old world, I will say, into the profile that I'm trying to get. I'm trying to get more into the French mineral, less fruity component. But with the wine that I'm working for I feel it was a good opportunity to play on a different profile, I will say, to not get exactly the same wine, and at the beginning was also just for me to have fun, to do something completely different. So that's the idea behind it, using the same fruit coming from the same vineyard and harvested at the same time, and make 2 wines that are at least trying to be slightly different.
Okay, great. So same fruit coming in off the vineyard, but 2 different styles, both just to see what the grapes are capable of, but also in this case to make 2 different wines because they go to 2 different labels. So to make sure they're different labels.
Yes.
So let's just take a look briefly. So you want to tell us a little bit about what your general approach is for the wine that you make in your old world style, and then we'll talk about the general approach that you used for the experimental wine, the protected wine.
I think it goes also with my style of winemaking. That, I mean, the direction I'm going to trying to use less product in my winemaking to be a little bit more hands-off. And therefore, I'm not trying to protect the wine from oxygen every single time I do something with it. I am, usually I've got a mix of like selected yeast and natural yeast. I think this one is only natural yeast, if I remember well. So it's also, I don't control the temperature on this wine. I mean, I do control the temperature for the clarification after pressing, but then after that, the wine goes in barrels, and it's 500 liters barrels, so bigger volumes. I'm not looking for oak impact, but I'm looking for the micro-oxygenation of the oak and the least contact during the fermentation and post-fermentation. I'm also working with a fairly low level of sulfur. That's got its advantage, but also its challenges. So that's things that describes this wine.
What about the next one? What did you do there?
The next one, I went exactly the opposite. So I've used all the products that I had in my toolbox that I could put in there. So it's much more intensive into enological product. It's also much more protected with from every movement being protected from oxygen with carbon dioxide. At least that's what I asked the guy to do. So if it's not done properly, that's BJ's problem.
It's done perfectly.
And so trying to protect the wine as much as possible of the oxygen, and use as much as I can to try to protect the style and what some people would consider as a Sauvignon Blanc character.
Okay, so we're going to kind of come back to this same figure a couple of times as we go through Mattieu's protocol. But before we do that, I wanted to just make sure that we have a quick review of the files so that we're all on the same page in terms of what he's trying to protect here and why we need to protect them. So, so the thials themselves, these are just reduced sulfur compound. These are sulfur compounds where it's a sulfur that's attached to an H, that's the thiol group itself. And then that's usually attached to a much bigger molecule. The ones that we're interested in for Sauvignon Blanc, and really for a number of other grape varieties as well, these are ones that we'll kind of call varietal thiols. These are ones that smell good. There's other thiol compounds that are reduced sulfur compounds that don't smell good, like our mercaptans, for example, things that can give us things like that are like garlic and rubber and onion, those sorts of things. Those are also thiols, and that's just because they have this SH group to them. When we think about the thiols in, oops. Are you guys seeing the thiol slide, or are you seeing something else? You're not seeing the thiol slide.
We're still on slide 4.
Oh, golly, sorry. Okay, we're gonna start over here, see if we can get back. Sorry for all of the difficulties here today. It's as if I haven't done this in a while. Okay, so do you see something that looks like thiols now? Okay,
Yes.
Sorry about that. Sorry about that. We'll see. Well, hopefully we'll keep moving forward. I'll maybe stop sharing periodically to reset it if we need to do that. But again, when we think about the thiols, then we're really thinking when they don't actually have this smell and aroma in your grapes. When you go out and taste your Sauvignon Blanc grapes, you're not tasting grapefruits and cat pee and that sort of thing. These are pretty much non-aromatic in their precursor form. So you need to have the precursors, but then once you also have to sort of have the enzymes in your fermentation in order to cleave off the larger molecule and give you the thiol itself. So these usually are being carried in on a cysteine or a serine, so some kind of amino acid. Sometimes they're also on a glutathione kind of residue. So you have to have that as well. But the other thing to remember about thiols is that they're very easily oxidized. So oxidation is basically when some molecule, usually something that has an oxygen involved in it, comes and steals that H away. And what happens when that happens is that it becomes non-aromatic again. So not only do you have to have the precursor to begin with. You have to have some means to transform that into its aromatic self. And then you have to be able to protect that from any oxidation that goes along the way. So the three molecules that we tend to think about with our varietal thiols are 4-MMP, which is that sort of boxwood or broom character. When you have it in high amounts, it can smell like cat pee. We also have 3-MH, which gives us, it's listed here as citrus, like limes and grapefruits and oranges. And to me, it always seems like a very sweet form of citrus. Like if you're going to like take that little bit of orange rind to put into a cocktail and you get that like spritz of the orange oil coming up, it's got that sweetness to it. That's how that presents to me, the 3-MH kind of molecules. And then the 3-MHA, that's actually a second step. So if you have 3-MH and it gets further modified by another enzyme, you can move into 3-MHA, which gets you more into passion fruit and gooseberry and that sort of thing. You will see that these are present at very, very low amounts. So they're measured in nanograms per liter. That's 10 to the -9 grams. So that's a very, 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 very tiny amount. And as a result of that, they really weren't known, they weren't detected for a very long time, because it is really hard to detect them at that small amount. But it turns out your nose can detect them. Our sensory threshold for these compounds is also very, very sensitive. And one other thing here is that we do need to also keep in mind that the thiols aren't working in their own. They also sort of interact with the other things in the matrix of the wine. And very particularly, they have been shown to increase our perception of other green characters, particularly methoxypyrazine, but also some of the hexanols and the green leafy kind of stuff. I will say we think about our thiols mostly with Sauvignon Blanc, but they have also been found in a number of other varieties. So they're in Chardonnay, they're in Céval, they're in your red wines. They tend to push to a different odor, a little bit different aromas that way. But they are present in a lot of other things. They're also very important in rosé wines. So even if you don't make Sauvignon Blanc, the principles we're talking about today in terms of protecting the thiols will still work in terms of some of those other varieties. So when we think about thiols themselves, we do need to remember, again, they come as precursors. And so if we're going to try to pump up those thiols, like Mattieu was talking about, some of that is making sure that we've got the precursors coming in. Some of it is making sure we're getting those thiol precursors from the fruit that's there. We also have to transform it, then we have to protect it. And there's a number of different things that have been shown to increase those processes. So in the vineyard, things like vine nutrition and leaf pulling will increase are supposed to increase your thiol precursors. Another one that does is berry damage. So if you have berry damage, apparently it triggers the plant to make more of these compounds. And that's, again, what's thought to be happening with botrytis as well, is that's sort of a plant response. Once you get them in,
having hail just before harvest is a good thing.
Perhaps hail before harvest as well. So
because that's what happened to this batch.
What we wish for, right? Exactly.
No, that's what happened to this batch. We had hail
really?
a week before harvest.
Well, it certainly has a lot of thiol, so maybe that's what it was. Maybe it's your winemaking. Once they come into the winery, there's lots of things that we can do to make sure to extract as many precursors as possible. So most of these are found in sort of the skin portion of the grapes. And so things that help us to extract from the skins are going to give us a little bit more precursor there, but we do need to be careful about part of that, which we'll talk about in a few minutes. The use of press enzymes can help with that. Additional press pressures can help with that. Another thing that would help with that is stabulation. So stabulation is just, instead of immediately cold settling your pulp, you kind of mix it anaerobically in order to extract more of the precursors from the pulp that's in the juice itself. But again, even if they're there, you have to have the enzymes to be able to cleave off that amino acid to release the aromatic portion. So that's where the yeast strain becomes very important. There are some yeast strains that express a lot more of those enzymes than others. And so you want to make sure to choose a yeast strain that's going to give you that expression if you're looking for it. Those yeast won't make those enzymes unless they have proper nutrition. So this is one of those cases where you do want to pay attention to feeding your yeast to make sure you have a good, healthy fermentation, but also to make sure they make the enzymes that are going to release your thiols. And then another thing is warmer temperatures. So temperatures between 18 and 20 have been shown to have higher thiol release or production than cooler temperatures. But again, once you have them, you've got to protect it, and that's predominantly has to do with limiting oxygen. So all of the things you're already doing in your cellar to limit oxygen, this is the place to be super careful about that. So being careful about gassing. Some of the protocols are like throwing dry ice in the press, throwing dry ice in the pan, gassing the lines, gassing your tanks, all of those things, keeping everything properly topped, judicious use of sulfur dioxide, and then also use of glutathione products. And we'll touch on a couple of those as we go into our protocol today. And then one last thing, it just is looking at how that oxygen is affecting the thiols, because again, that's going to tell us what these different components of Mattieu's protocol were doing for us. So it turns out when oxygen comes into your wine, molecular oxygen as it dissolves into your wine really isn't that reactive with everything. But even if there's a little bit of a metal catalyst around, it's going to turn that oxygen into a reactive oxygen species. There's lots of different forms of this, but just generally those are things that are going to be more sort of more reactive within the wine matrix itself. Taken to its most radical form, you can get to a hydroxide radical, and that's what's really going to be the most potent oxidizer that you'll have. And that's just going to basically oxidize anything it can find, and the easiest thing it can find, most of which is going to be ethanol, because that's going to be the first thing it bumps into. So when ethanol gets oxidized, that's when you get that sort of aldehydic affect to the wine. Other things like acids and sugars can also be oxidized into keto acids and aldehydes. So that's sort of that nutty, bruised apple kind of character that the wine can take. That's sort of part of where that's coming from. But the reactive oxygen species then can also go and interact with phenolics. And we might not think about our white wines having a lot of phenolics, but the pulp in your white wine has a lot of hydroxycinnamic acids. That is a type of phenolic, and those are subject to these particular reactions. Once again, this doesn't really happen a lot on its own. You do need catalysts to have that happen. So the most prevalent catalyst comes from the grapes itself. So we can't really do anything about making that not be there. That's polyphenol oxidase. I will say that if you have botrytis infected fruit, there's another enzyme called lacase that will also do that conversion, and it does it more. It is harder to inactivate, and that can cause a real oxidative problem. Iron can also be a catalyst at this point.
So can copper.
So can copper. Yes, thank you, Bruce. So these are kind of, I'm using iron as a stand-in. In a lot of cases generally for metals, but yeah, copper can definitely, and especially if you're spraying copper in the vineyard, that's something to think about. But what you end up here with is a quinone, and the quinone is really what's going to be causing the problems for us. So the quinone, if there's glutathione around, the favorite thing for the quinone to bind to is glutathione, forming GRP, which is a grape reactive product. The grape reactive product basically has no smell or no flavor, nothing. That's not going to cause a problem for us. So that's like the good thing. That's where we want the quinone to go if we have it. Unfortunately, that does use up glutathione over time. And once you run out of glutathione, the quinone finds other things to bind to. And predominantly, that's going to be our aromatic compounds, our thiols, our esters, our terpenes. And it's also going to find some other phenolics around that's going to cause those phenolics to brown and potentially give us some bitterness. So when we think about that oxygen coming in, this pathway gives us lots of different places where we might be able to intervene to try to keep things from progressing down to this portion where we're actually oxidizing our aromatic compounds and leading to browning or bitterness. Okay, so with that in mind, let's jump back into Mattieu's protocol here. So Mattieu, we're just going to kind of go step by step through your protocol and talk about why you did each one of these things, and then we'll see what effect that had on the wines. So when you were pressing, you hand harvested, you whole cluster pressed, and you diverted the press fraction. So is that just your standard procedure, or is there a reason you were doing that on your Sauvignon Blanc?
No, it's just a standard procedure. So yeah, I'd rather. I'm always concerned about the acid, and I know the more we press, the more the juice that we get at the end. Even if technically they might have more sial, they also will get more bitterness, more phenol, and higher pH, and that's not what I'm looking for. So I try to separate my press fraction. Usually it's I keep the two first third for the varietal, and the last third goes into a different one.
Yeah. So as Mattieu said, some of that is about pH, but some of that also, if you don't have as much phenolic around because you haven't pressed as hard or you haven't kept that press fraction, you just have fewer things to kind of go down this left-hand side of the diagram. But we do see one difference here. In your normal protocol, you do not use any pressing enzyme. And in this case, you did use pressing enzyme. So do you want to talk a little bit about what you were trying to get from the enzyme, or why you used enzyme here and not in the other one?
Well, because it tells you that it's a good way to extract sial and flavor precursor. So, it also helps with clarification, but it's on this specific case is mainly to try to extract sial.
Yeah, so again, this is just one of a bit here, but again, those precursors are in the skins and you're not like pressing too hard. So that's part of it. Okay, so then you pressed into tank, and in both protocols, you held in the tank for 5 days. Do you want to talk a little bit about why you're holding in tank for that long? What's going on during that time?
So the first thing is I've got other shit to do. I'm always trying to ferment after I press. So sometimes it's easier to just keep it in the tank cold for 5 days when I've got time to deal with it. That could happen. But also I did experiment for a while with stabilization, and I really like stabilization. So I realized that leaving your juice in a tank for 5 days, it's more beneficial than not. So I'm less worried about racking it the day after. When I start doing winemaking, I was always cold setting overnight, rack it the next day, start your fermentation. I was always worried that the leaves gonna start fermenting and I'm gonna have off flavor. I realized that it doesn't happen that much anymore, and I think the stabilization really helped for the aromatic profile of the wine ultimately. But for my regular Sauvignon Blanc, during that time, I'm also getting some juice that I'm starting my starter in buckets outside the winery. And so I need this time to get my starter fully started before I can rack it off and then inoculate with my starter.
Great. So it's actually serving two different purposes there.
Right.
I would say we do have a number of previous trials on stabulation, and it does seem to have a pretty profound effect in pumping up those thiles if you can get them. Okay, so from there, then you racked into barrels for your standard protocol, and you went into tank for your other protocol. You talked about that just a little bit. Is there anything else you want to kind of add about your vessel choice there?
Again, to try to preserve the sial, I want a stainless steel fermentation, or at least something where I can control the temperature a little bit more. I know that the sial are usually extracted more on 20 Celsius, so 68, 70. That's usually where you extract the sial. But I think, at least of what I've read about it, it's better to start your fermentation a little bit warm for the sial extraction, and then after 1/3 of fermentation, bring it down to more like your classic 15 Celsius or below. But it's better at the beginning to be more around 18, 20 Celsius. Somebody wants to do translation in Fahrenheit, I'll let you do that. I don't speak Fahrenheit.
Great. And we did have a question come into the chat. I'm sorry, did you want to talk about barrels first? I didn't mean to cut you off.
And the barrels, it's not only the barrels, I've got the micro-oxidation, but I've got temperature that's going to stay a little bit higher as the entire time. I mean, I'm more like 18, 20 Celsius during the fermentation. But I also have I don't want to rack it off after that. So I ferment in barrels and then let it sit in barrels until I'm ready to bottle this wine. So it's it's a bit more of this kind of like hands-off approach about this wine. And it's a 500 liter barrels. I also realized that you put that my NTU was at 60. I've been on both of them, I think, adjusted my NTU level, I think close to 150, at least 60 will be too low for me to start the fermentation. So I try to get my NTU level between 150 and 200 when I work on the Sauvignon Blanc. And I will say for the regular Sauvignon Blanc, for the modern style Sauvignon Blanc, I'm probably my target was probably more 240s than 200.
Okay. Yeah, this may have been what the NTU was before you measure, like in the first measurement, yes. And then it was probably corrected. This is just what ended up in the yes. system. But thank you for clarifying that. So we had a couple questions coming in. The temperature at crush. I think probably these were the grapes were chilled overnight and then crushed the next day. Is that?
Yes, this year, yes. This year I was able to get it cold. Last year we had a problem, but that's a different subject. But yeah, this year the temperature was cold room. So cold room temperature, send it to the press. We do a sorting before we get to the press. It's the press that we use for this wine is a closed tank press. It doesn't matter anyway, but like it doesn't matter for my personal style, but I think for the more reductive style, it's much easier to control the oxidation out of it.
And then somebody was also asking about some fermentation kinetics. We'll have the pictures of that in just a minute. And I should also say, I just dropped into the chat the draft report for this. So it just gives you a little bit more of the rates for things, but you'll also see the raw data for things. So if you're interested in those, we can go ahead and do that. Okay, so let's start talking about this. You sort of have this big list of stuff that you added to your protected version that you didn't add in your permissive version. So let's talk about what some of these things are doing. So the first one that you added was optithiols. What was the optithiols for?
It's a cereal precursor to help something. It's a yeast byproduct rich in glutathione, and so that does help to protect and extract the cereal. Again, like I'm not sure exactly what it does. It just says optithiol. So I'm like,
so yeah, if we look at the product literature, and I'm just telling you the product literature, and I should say here too, like we're telling you the exact product that Mattieu used, but if you are a Scott Labs person, Scott Labs most likely has a version of this. If you're an AEB person or a Lafort person, we'll see some Lafort products coming up. Talk to your favorite supplier if you're interested in these, and they probably have their own version of this. So the optithiols is sort of, as Mattieu said, it's sort of that version of the specialized nutrient that's supposed to pump your thiols. So it's bringing in those precursors. You have to add it really early in fermentation to make sure that it gets that enzymatic action. But some of those precursors are hitched to glutathione. So when you cleave them, you actually also release some glutathione. One thing that I did read as I was looking into this is that the glutathione has that sort of good protective thing later on, but it also turns out that early in fermentation, it can be used by the yeast to convert some toxic C6 compounds, which are toxic to the yeast, but also produces then glutathione hooked to 3-MH that then gets cleaved off to make more 3-MH. So adding this product early on, you kind of get that potential benefit along the way. Okay, so that was the first thing. The second thing was that you did use, in this case, you used X5. Yeah, so you used X5 yeast. So you've used this yeast before. Tell us about why you chose this yeast.
We used it a couple of years ago in my classic style of Sauvignon Blanc in barrel fermentation, and compared to my natural yeast fermentation. And it always gave me this very a Sauvignon Blanc component, like this very new world component. So I feel like it's a yeast that I'm used to, and I think it's a good candidate for me to use it to try to get the cereal.
Yeah. So as Mattieu said, we actually did do a comparison of this a couple of years ago, just in barrel fermentations. The only thing being different in the yeast group And we did see significantly higher scores for citrus and boxwood and tropical fruit and green character and complexity for the X5. And so it does seem, again, the product literature will say it has the enzymes that we need for that. The code word in your product catalog is always either thiol producing or recommended for Sauvignon Blanc. Sometimes they'll actually name the enzymes as beta lyase. But if you're curious, again, your supplier can help you to find the right category for whomever you're purchasing your supplies from. Now, I noticed, Mattieu, that you used yeast rehydration nutrient, and that's not something that you always use. So why did you use the rehydration nutrient this time around?
In fact, I'm using more and more. I used to use GoFirm. That's exactly the same product as GoFirm. It's just I find it's much easier to dissolve than GoFirm. It's supposed to do the same, and I find it much easier to dissolve than GoFirm. So that's the only reason why I'm using the Steam. But the more it goes, the less I'm using nutrient during my fermentation. Even when I use selected yeast, because I figure that I realize that usually I don't have a lot of problem with reduction of flavor fermentation or sluggish fermentation. But I do, if I do inoculate with yeast, I do like to be sure that my yeast start on a good note, and I do use some yeast rehydration nutrient.
And one thing too with this is that, when you're being really reductive in your winemaking, you also have the potential to kind of not get rid of any other reductive aromas that might be around. So one of the arguments for good yeast nutrition from hydration all the way on is just not putting your yeast in a situation where it would produce H2S or any of those sorts of things. And the rehydration nutrient is a piece of that. So, okay, so you also used, I can't see all of my thing is in the way. There we go. Laphazyme thiol. So this is an enzyme. You want to tell us a little bit about the Laphazyme thiol?
So that's I started using this product on my rosé when I was doing the stabilization experimentation. It's something that you use at technically, if you do a true stabilization of almost more than 1 week at cold temperature, you should get exactly the same result. But they were saying that with this enzyme, you will get the thiol extraction a little bit faster. So it did at the time on my trial for the rosé, it did work very well. So here it is. I did on.
And this was one that was featured in the Lafort trials in I think it was 2016. Bruce was a part of that. There's some really good information online on this particular enzyme. And so I do encourage you to kind of take a look at that information if you're interested. But one of the things that stood out to me is that at least in some of those trials, the enzyme had more effect than the stabulation itself did. So, but the product literature does say it does not make up for not having a good strain of yeast. So don't think that you're just going to get all the enzyme you need from the addition. It's not enough to do that. So, okay, what do we have next?
I would say that on these thiol-producing yeast, particularly the Lafort products, you need to very much pay attention to the nitrogen level. Your one slide had something about moderate nitrogen consumer. If the nitrogen level is low, the results are not as favorable. We found that out with some of these early reviews that we did at Lafort.
Great, thank you. And yeah, that's actually because I think probably because of the work that you all did with that, that's included in all of these. Any of the protocols for this are always talk about proper yeast nutrition. So we'll talk about Mattieu's nutrition in just a second, but that's a great point. So Mattieu, you also used Fresh Aroma. So tell us about Fresh Aroma.
It's a glutathione product that's been on the market for a while now. And I think it's, using it at 1/3 of fermentation helps to protect the wine from oxidation again. About everything that we said about the glutathione, but again, just to get back, this whole experimentation was fun because I had to put everything that I had in my toolbox, So everything that's acyl went into the wine, and everything that was supposed to protect me from the oxygen also went to the wine. So there's a lot of things that have been added that I will usually not combine all of them at the same time. But
Right. Yeah. So, I mean, that's a good point too, that like we sort of originally talked about just putting enough precursors in there that you have the thiol, but now we're starting to move into the protecting it to make sure you keep it. And again, adding these thiol, these are really kind of thiol precursors that come in, but the literature talks about it like it gets into the yeast body, and then when the yeast body dies, it starts to like give it back up. So it's almost a little time release capsule for glutathione. So when you've used up the stuff that comes in on your grapes, your yeast start giving up this glutathione back to you. And again, this is just looking back at this, at that very end product to try to quench that quinone so that you don't get the other things attacked. We also did have proper, as Bruce brought up before, this did receive yeast nutrients. Mattieu doesn't always use yeast nutrients, but in this case, you did use proper yeast nutrition. I would say, as Bruce brought up as well, paying attention to what the nutrition needs are for your yeast is important, and feeding appropriately. There was one part of the literature that did also say that the expression of the enzymes for thiol conversion can actually be turned off if your nitrogen is too high. So this is not the case where you just feed it as much fermented K as you're legally allowed and hope you're okay. Like, you do need to actually measure your YAN level and keep in mind the nutrient need of your yeast and feed appropriately for that. You don't want to overdo it in this case. Okay, so how are we doing? We have gotten sort of through the portion where we kind of we brought in the precursors, we made sure we got them, and we started to protect them. But then, Mattieu, you also added some fining agents. And these fining agents are really ones that the shift here is just to take out things that might be hurting things. So do you want to tell us, the first one you used was casein. What was the casein for?
It's to remove oxidative components and prevent any phenol that can be oxidized later. The finding bentocasein during fermentation, during alcoholic fermentation, it's something that I've done. It's one of my old habits that I've had when I started making wine 25 years ago, and I haven't really changed it since. Every time I do a white wine in tank, I will use bentocasein during the fermentation. 40-20 usually, I think 40 grams per hectoliters and 20 grams of per hectoliters. It's one thing that I don't think too much about doing it. I'm just do it. 1/3 of fermentation, I do add the bentocasein, and that's it. Also, Sauvignon Blanc has a lot of protein to start with. So we've done a lot of trial with protein finding, and there's different school and different people do different things, but I like to do my finding earlier than later if I can. It will allows me to, even if I do find it later on, to add a little bit less bentonite.
So in the bentonite, the bentonite, as you said, like sort of helps with that protein finding early on, theoretically before your aromatics are released. It's also something that some people will use to remove polyphenol oxidases. So those are a protein, and remember, that's one of the catalysts along our oxidative pathway. But one thing also that I read along the way was it can also remove some glutathione, and bentonite can contain iron. So you want to look for a bentonite that is a low iron bentonite, and just sort of watch those glutathione levels, realizing that you might be taking some of that out along the way. And then you use Clairol HM. This is PVPP and ketosan. You want to talk about that for a second?
Yeah, I mean, the main reason why I've used it is because it absorbs heavy metal. It is a product that I start using with my canning. So I do put wine in cans, and reduction in cans is a big problem, especially because if you got a bit of copper, combine copper into your wine, that can really link to like very heavy reduction later on into when you can, especially when you got reaction with the aluminum from the can. So to try to avoid to keep, when you do canning, you need to have your almost no free sulfur at the time of canning, and to try to remove the chance of oxidation. when you work with very low level of sulfur, Claryl HM help to keep the freshness in the wine that you can by removing mainly the copper bond sulfide.
So if we look at sort of the oxidative pathway, then again, your casein is sort of taking out some phenolics. Your bentonite is helping with the PPO. The Claryl HM is helping with some of those heavy metals. So you're sort of, again, just chipping away at these different ways that you might be moving down that pathway to try to sort of move that pathway a little bit less. Mattieu, we do have a question. Shai is asking if the bentonite was the specialized fermentation bentonite, or if you're just using a standard bentonite. Do you just use a standard bentonite?
Yeah, just the cheapest that I can find.
And we know which one that is. Okay, so we're almost done. The last thing we need to talk about is sulfur dioxide. So you have said that you try to work with sort of low sulfur dioxide in your regular winemaking, and a lot of these interventions are ones that can help to sort of make it so you don't have to use as much sulfur dioxide. But when we just sort of think about where SO2 is acting on our oxidative pathway, it turns out that SO2 does really help with a lot of these different things. So, SO2 can also work against your polyphenol oxidase and slow down that reaction. When you do have some of these hydrogen radicals or these hydroxide radicals, it will help to quench those. When you do get some acetaldehyde, the SO2 is going to bind to the acetaldehyde, and it's going to make it not have as much flavor or aroma or flavor to it. And then it also can help to quench some of the quinones. So just judicious use of SO2 is important here, even if you're trying to kind of reduce that. If you're trying to keep your thiols, you do need to kind of at least consider using some SO2, and Mattieu did in this case. Okay, so you guys have been very patient. Let's figure out what's in the glasses in front of you and talk about the experiment itself. So if you're in group 1, I'm calling Protective is going to be that sort of more reduced one that has all of the additions to it, and Permissive is Mattieu's normal protocol. So if you're in your group 1, numbers 614 and 427 were both Protective, whereas 242 was Permissive. If you're in group 2, wine 902 was Protective, and wines 503 and 856 were Permissive. Group 3, it's 206 and 884 were Permissive, and 525 was Protective. And if you're in group 4, 789 is Permissive, and 167 and 353 were Protective. And Jenna, can I have you just double check that with the flight key? That I think there might be one of those that might not be correct.
I'm group 4, and I don't think that's what it is. But
yeah, and I'm in group 2, and I think it's flipped. I think I have them all flipped. So Jenna, if you don't mind checking, we changed the verbiage here along the way. So I mean,
yeah, so if that's what it is on group 4, I did something wrong.
Yeah.
With exactly
I'm thinking that they're all opposite, and that is absolutely my mistake there. Jenna, but like I said, Jenna's checking the flight key for us. So let me stop sharing here. I think it's because we changed the names of them along the way. So. Somebody says it's better. Let's taste them blind. Yeah. So Jenna, if you have that, if you can reread the answers there sure. With the like I said, we just changed the verbiage. So I think.
Would you like me to share my screen? Would that be easier?
Yes, that would be great. Okay.
No, I still don't think it's right. Like the group 4, I've got group 4, like, I don't think the 789 is the oxidated one. You can see it on color also, like the color, that you can see the difference on color. And I don't think that.
This is fun. So can we have, Jenna, if you want to stop sharing your screen for a minute. If we can use the raise your hand function. So if you want to, we're just going to do a quick raise your hand. So if you were able to tell the difference between the wines, go ahead and raise your hand. So for those of you able to tell the difference between the wines, what was different for the wines? Yeah, AJ's saying the color difference is pretty evident. I felt like the color difference was pretty clear. So if you can put your hand down for just a minute. If you were able to tell the difference between the wines, did they seem did they seem opposite to you? Yeah. I think we maybe had
Yeah.
I think we have a labeling problem somewhere in the wine.
We'll
somewhere
blame
along the way.
DJ here, we'll blame him here for the sampling, but okay.
Yeah, so I think we have a labeling problem somewhere along the way there. So we will track that down. But let's go ahead and discuss the wines as if they were opposite, because I think that is most likely what's happening here. So flip around the key that I gave you before. What differences did you find in the wines besides the color? So we know that the color was different, but what other differences could you find in the wines? Because we seem pretty like convinced that the key is opposite. What else about them? So we've got Shai, if you could be specific, so which one had more weight or less weight, which one had more acid intensity, which one had more fruit intensity? Tell us which side of things those were weighing out on. So Tom says more opulent, clean fruit in the protective lot. There. Melanie says 242 was super thiol-y. So 242 we're in the flipped version is protective. So so John says 242 much more protective, aromatic, and brighter fruit. So again, 242 is going to be that protective one.
That would be the same as the 789.
Yes, that would, yes. 789 has brighter fruit. Permissive was sweeter citrus tropical for Melanie. So she felt like the protected was super thiol-y, but the other was sweeter citrus tropical, so a little bit more shifted to the tropical. So 884 and 206. So again, our protected was tropical bombs, where the other was like a dull barrel fermented. So it had less of that varietal character that you were looking for. Rick knew it was
I wasn't looking for that varietal character on the other one, so it's great.
Okay. Okay, so let's try this. So I have a poll question. We can go here. Everyone does have to vote. Let's see. I would like to know which wine you preferred. Okay, so which of these wines would you rather drink yourself? Or make? I guess you could say, which one would you rather make? We have just a couple more people to vote here. So if you haven't voted, please do. Just a few more seconds here to vote. Which one do you prefer? Would you rather drink or would you rather make? Okay, let me go ahead and end that. And I am going to share the results. I hope you guys can see these when you see them. Can you guys see the results of the poll? No.
Yes, we
Some
do.
are saying
We
yes,
do, we
and
do.
some are
Yes, we
saying
do.
yes, and some are saying no. So can you see the results? Yes.
Yes.
Okay. I think everybody but Jenna can. I think anybody who's not a co-host can see them. So if we look at this, we have 56% of the voters preferred the permissive oxygen treatment, and 44% preferred the protective oxygen treatment. So let's talk about that a little bit. Let's talk about if you were one of the people who preferred the permissive oxygen treatment, what did you prefer about that wine? So John says that he agreed. Normally, he prefers the New Zealand, but that was kind of too much. So it was sort of like all of the things just went too far. Okay.
Correct, yeah. The oxidation definitely improved the palate, and I did appreciate the weight that it garnered, but I would generally, for Sauvignon Blanc, I want to see more aromatics. So it's somewhere probably in between for me.
Yeah. Yeah. And we didn't talk very much about that. So thank you for bringing that up, John. I felt like the palate weight for me was more like I felt like it had more weight to it in the permissive treatment than in the protective treatment. Debbie Schick says full body tropical fruit. So the full body part of that sort of setting apart the permissive style. Anybody else? What else did you prefer about the permissive style? Okay, what about the protective people? Those of you that prefer the protective style, what was it about the protective style that you liked? I think Rick, this is probably still that last one. So he liked the talky minerality of the permissive style. So for the protective style, Joyce likes at first she liked the New Zealand, but a few glasses into a bottle, it would be tiresome. So basically she prefers the French style overall. I think that's an important.
On the long run, it's always better.
That's a good point, though. Sometimes too much becomes too much, right?
I think my boss here at Box would like my answer too. To focus on French style, the Bordeaux style wines too.
Okay, well then you're well placed, right?
Right.
I mean, I think what's interesting is like what I found interesting with these two wines is like with the same grapes, you can really go two different ways. And with two different styles, and like you see, like some people's gonna like the fruitier, brighter style much better, and sometimes the more restrained style. I've got a lot of people that tell me they don't like to drink Sauvignon Blanc because it's too fruity. So you know, like the same kind of people that would tell you they don't like to drink Chardonnay because it's too oaky. So it's there's always a world in between. Also, one thing to justify the more oxidative style winemaking is I'm planning to do a much longer aging on that. So it's like with the oxidative style will be bottled next week, in 2 weeks, while the reductive style will be bottled in 2 weeks. The oxidative style will be bottled next year. So it's not the same. I'm not looking at all for the same characteristic here.
Yeah, and I mean, if you were looking for that, I mean, you couldn't age it that long, otherwise you would lose those styles in the course of aging anyway.
Exactly. And the other one, I already don't have much styles, so I don't care about them anymore. I don't have to, you know, protect them.
Yeah.
It is a really interesting question, because the reality is we are not producing for ourselves. We're producing for consumers, and not just consumers that you're going to present the wine to in the next month, but it's the next year. And that is the one problem I've had with my really aromatic whites, is that they're dying. You know, after 6 months, 8 months, they start to really go off a cliff, and the reductive styles definitely stick around.
And I think, John, that's a good point too. We didn't really deal with any of the things that happen after this. So cold stabilization, you bring up a lot of oxygen. Every time you're racking the wine, when you're preparing for bottling, when you're filtering, when you're getting in the bottle, there's a lot of oxygen ingress that happens during that time. So even though it might feel like it's too much on this side of it, if you want to still have that aromatic wine and the other, by the time you get all the way through the bottling process and even some age in the bottle, it might be that you need to sort of pump that up here. So you've got you've got more to begin with. And again, that glutathione and adequate SO2 are protecting them a little bit.
So
We have
Well,
a really-
that oxygen protection is one of the reasons why the New Zealanders and Australians were so big on ascorbic acid, which never has really caught on here, primarily because of the problem of proto-oxidation. That is to say, if you don't have the right balance of sulfur dioxide and ascorbic acid can actually increase the rate of browning, which kind of freaks out most American winemakers. But the people that really have it together use it and it's a much more viable antioxidant than sulfur dioxide by a country mile, and even more so than glutathione.
Yeah,
there's
that's a good point.
also at bottling some tools that you can use. So the first, like if you do a cross-flow filter, you will have less chance of oxidation than if you use a plate filter when you filter your wine. If you be sure that you don't have any dissolved oxygen intake during your bottling and your filtration, that's also something very important. And one of the things that I do very often for this more reductive style winemaking, I use some tannins at bottling. There's a kind of tannin that's called like tan SLI that I'm using from an artist. That same thing, it's an oxygen scavenger that you can use just at bottling. And even if you're, you can bottle with level of sulfur that are more in line of what you're targeting, and without, limiting the oxidation, the extend the shelf life of your product, I would say.
There's a really interesting comment that came in from Ben Sedlins. He said, tasting blind, I preferred the New Zealand approach, but knowing all the differences in the winemaking, i.e. all the additives, makes me reconsider. And I think that is something to keep in mind too. I wondered as I was writing out this protocol, like every single one of these has a rehydration step. Every single one of these things you're kind of adding and you're pumping over and you're moving and that sort of thing. Each one of these you're purchasing separately. And I do think there is that sort of consideration of just how many things are going in there. I thought that was a very interesting comment.
Yeah, if you're a lazy winemaker, it's better to do the oxidative styles than the reductive style.
Yeah, not exactly minimal winemaking.
Yeah, exactly. So, okay, well, I realize we've gone over time just a little bit, and I do want to honor folks' time. Just a couple things as we wrap up, and then I'll say, I'm happy to stay on, and if Mattieu can stay on, if there's additional comments or questions, we'd love to take those. We do want to make sure to thank Mattieu and King family for doing this experiment in the middle of harvest and sharing that with us. We always
my
want
team
to
here. They're the one doing everything. I'm not doing anything. I just give the write down work orders.
Those are the guys that had to add all that stuff. We also need to make sure to thank the Virginia Wine Board for their continued funding, and thanks to each one of you for filling out your sheets and coming and participating in the discussion today. And like I said, we will have our next sensory session is in 2 weeks. You should see that email hit your box at the end of the day today. And if you don't we'll kind of send out another email to say, if you're not seeing the emails, try these kinds of things, and we'll get that figured out. So we're sorry for all of the technical issues today. Thanks for sticking with us. It's great to see each one of you, and we'll see you again soon.
Thank you for putting that on, Dr. Ting.
You're welcome.
Yeah,
Yes, and
good
happy
job.
birthday.
Thanks, Mattieu.
You're welcome.
So Bruce, I think you really make a good point about the ascorbic acid. Like I didn't even include it. I mean, I didn't include it because nobody I know actually uses it. I don't know if anybody here is using it, but I think you're right.
It will,
Kind of that frees you out.
I have some West Coast clients that use it, but I know when I used to chat it up here in Virginia, I didn't get too many takers because they'd look at the literature and you can really run into some problems if you don't manage your sulfur dioxide level properly.
Yeah, I think you need to be around 35 ppm of free sulfur when you bottle. No, that's what they recommend to be.
That high?
Yeah.
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 So that's the reason why I didn't use it.
Yeah.
Yeah. Well, it depends on who you believe. That's what Delte says and some of the other people that we brought in here. But I have got folks I know that didn't go anywhere near that high.
Well, if anybody would like to experiment with that next year, I'm happy to talk to you about it. We can bring in Bruce as a consultant to make sure that we're doing it right. So
I'd like to experiment with it right now. Try to stave off my oxidation in bottles over time.
yeah.
Well, I have on my analogy notes, there's a couple of postings on the use of ascorbic and some of the concerns. So you can at least find out what we've tried and what's worked here in Virginia and what hasn't. It's at www.vtwines.info, info. And just click on enology notes and it's under the index under ascorbic acid. Or call me if you have any issues, if I can help.
Thank you. That's maybe.
Write up the plan and then email him to see what he thinks.
No, I have, I'm thinking I got 18 pallets of Viognier to fill, and I could easily mark off some of the latter ones and just doing a ascorbic trail in there.
Yeah,
Yeah.
it's a good option.
They definitely, they just go off a cliff. It's a steep curve. So spectacular now, but then it's like 6 months, 7, 8.
Yeah, that's not an uncommon problem, unfortunately.
Bruce, do you feel like some glutathione might be helpful there too?
Sure. I think you brought up a good point on measuring the metals. Got to have the metals under control, otherwise it doesn't make any difference what antioxidants you have. Because of that Fenton reaction, which I mentioned is not just for iron,
Right.
even though that's how it was named.
Right. Yeah.
There's really some good, not that I'm advocating for them because I'm trying to get away of using too many things, but like the Claryl HM is one of them, but everybody's got some finding agent that really remove some of the shellac metal, and that does work very well. I've done an experimentation between 2 batches of rosé 1 year, and it was very interesting to see the difference of flavor just with the finding agent, just by removing the metal.
I think all of the research that's being done, Mattieu, as you brought this up on sort of how to keep our canned wines from going reductive, I think a lot of that also has to do with some of that sort of, I think it sort of pushed some of the metals material like more to the forefront because people are thinking about it more carefully now, especially because of the
Well, that's right. Plus you're lowering that redox potential so dramatically that you can get a lot of things springing out that weren't evident before in their oxidized state, but as soon as they get in a reductive state, the sensory threshold is so much dramatically lower.
Yeah. So there is a question in the chat about the URL again. I would just say that for everybody, all I ever do is I do a Google search for enology notes, and it's the first thing that comes up, and then you get this repository of great information from Bruce.
Yeah, they changed. Thank you. They changed the tech has to change servers and everything else pretty often. So sometimes a little confusing, but
yeah, I just always go with whatever, like I said, the Google search, it's the first thing that comes up, and it indexes very well through Google, so it'll always show up. But I just did also throw that into the chat as well. And if you haven't found enology notes before, you're in for a treat, because there's something, Bruce just has many years of wisdom to share through that written resource. So thank you, Bruce, for doing that for all those years for us. So.
You're welcome.
And Rick is holding up his sparkling wine production, and that Bruce has like a textbook on sparkling wine production that I think was fairly helpful for Rick in the last couple of weeks. So there you go. So,
good.
Yeah. Okay. Anybody else? Any other comments or questions that that might come up that you're interested to talk to our experts while they're here?